In the last video about cancer, we talked about some general ideas about how cell growth and cell death are regulated, and in particular, we used the fairly solid but simple model of how there's a balance between cells that are capable of dividing but are quiescent, that is, that aren't dividing, and under the right circumstances, those cells being triggered to divide, then after division, returning to a quiescent state when there are enough cells, and how this program of regulating cell division is balanced with a set of signaling, opposing signaling programs that trigger programmed cell death or apoptosis, and in fact how many of the same molecules regulate both of these processes, and how cell division can happen until you have too many cells, or if a cell damages or it detects DNA damage, or if there's no signals telling it to grow, then a variety of signals can induce apoptosis in cells that have finished dividing. They can also trigger programmed cell death in a quiescent cell. What we haven't talked about really very much is what is the role of RNA processing, and in particular RNA splicing, in not just triggering cell division or apoptosis, but in controlling the balance between growth and death. And that's going to be the subject of this video. We're going to start by talking about one specific example. So some background. It turns out that the process of triggering cell division or allowing cell division, and the flip side of that, triggering apoptosis or triggering cell death, are really sort of two sides of the same coin, and they're regulated by many of the same proteins when things can go either way. One of those key linchpin proteins, and not coincidentally a protein that is frequently mutated in cancer cells, is a protein called p53. p53, shown here, is highly receptive to lots and lots of different kinds of signals that indicate abnormality. They can be signals that are received through proteins, through signal transduction, etc. They can simply be a response to things like DNA damage that, if it's present, would be an indication the cell probably should not go on and divide. When p53 becomes stabilized, it interacts with other cell cycle proteins and blocks one or more of those different cell cycle checkpoints and arrests the cell. And it will, rather than just saying, okay, die, it will stop cell division and give the cell a chance to fix itself. Under normal circumstances, when everything is A-OK in the cell, p53 will be complex with a protein called MDM2, and I'm going into lots of detail now because we're going to see this again. And that leads to something called ubiquitination, which is the addition of tiny proteins called ubiquitin to p53, which targets p53 for destruction. In a normal dividing cell, p53 is constantly being made and destroyed as fast as it's made, so that it's there to respond and stop cell division if that needs to happen. Previously, we saw a more complicated, more busy, more encyclopedic version of this figure that showed that there are many genes that can be mutated in cancer, and especially when we consider the many genes that are only found with fairly low frequency in tumors. It turns out that a number of those relatively low frequency mutated genes are genes that their normal function is to regulate splicing. Now, these aren't genes who have a specific role in regulating only cancer genes. These are proteins whose normal function is to regulate the alternative splicing of lots and lots of different kinds of genes that have many varied effects on the cell. But because some of the genes they regulate through alternative splicing are genes that are involved in cell division and apoptosis, these can serve to drive cell division. And in fact, if we look on this list of genes that are globally effective in regulating alternative splicing, we find the gene RBM10, which we know is an interesting regulator of alternative splicing. One of the genes that is regulated by RBM10 is NUM, and I think NUM serves as a good sort of poster child and illustration for how alternative splicing can lead to the regulation of the balance between cell division and apoptosis. Genes like NUM that have this kind of a role are called pro-apoptotic genes because their activity can, when it's present, can help the cell to be more capable of making the choice to start programmed cell death when things are defective. It turns out that when you look in cells, there are 4 primary forms of the NUM protein that are produced in those cells. And those proteins come from 2 different alternative splicing events. That is, whether or not you include exon 3 or whether or not you include exon 9. And those 2 alternative splicing events can lead to 4 different proteins because you see combinations of proteins that have exon 3 or not and have exon 9 or not. You see all 4 different possible combinations. Why does this alternative splicing have an effect on cell growth? Remember our friend MDM2. MDM2 is this ubiquitin ligase that binds to p53, ubiquitinates it, and that targets p53 for destruction, which allows the cell to divide. So MDM2 is promoting cell division. It turns out that MDM2 itself is in turn regulated by NUM. And not just by anything about NUM, but it's bound by 11 amino acids that are found in exon 3 of the NUM protein. So if exon 3 is included in the NUM protein, it binds to MDM2. That prevents MDM2 from grabbing onto p53, and in turn that allows p53 to promote cell cycle arrest and apoptosis. So as long as exon 3 is there, NUM can function to allow p53 to do its job. Therefore, it makes p53 more pro-apoptotic. Skipping exon 3 then strips NUM of its ability to bind to MDM2, and it allows MDM2 to remove p53 and promote cell division. So before I go into a very confident sounding explanation, I think it's fair to say that nobody really understands 100% what NUM protein does, and particularly that applies to exon 9. It's more a question of correlations here. But here's what we think we know. So there's another protein called Notch. Notch is a family of proteins actually that are cell surface proteins that represent their own distinct kind of signaling pathway. When a Notch protein is bound by a ligand on another cell or a soluble ligand, it triggers a very bizarre kind of signal transduction. The Notch protein itself gets cleaved and it releases its cytoplasmic domain. In the case of Notch, the most common thing that happens then is that cytoplasmic domain goes into the nucleus and activates the transcription of genes that promote cell division. So Notch signaling promotes cell division. So it turns out that whether or not you include exon 9 in the NUM protein influences whether or not NUM can inhibit Notch signaling. When you add the sequences from exon 9, it seems like it disrupts the ability of NUM to bind to this NICD. But when you skip exon 9, you make a version of NUM that can bind NICD. It prevents it from entering the nucleus, and it blocks this part of Notch signaling that stimulates cell division. Now, there's studies that suggest that the full-length NUM protein, including exon 9, might antagonize, might bind to and inhibit the ability of the exon 9 skip protein to do this pro-apoptotic function. And there are studies that suggest that, well, maybe NUM has something to do with the function of Notch in endocytic vesicles, and that NUM may play a role in regulating all of this through endocytosis. And we don't really know everything about what NUM does because it's a complicated protein. But what we do know is that if you skip exon 9, you make a version of NUM that can block Notch signaling. When you include exon 3, you can make a version of NUM that blocks MDM from promoting the ubiquitination of p53. And in turn, RBM10 regulates the splicing of exon 9 of NUM. So we know that from the last slide that if you skip exon 9, you make a version that inhibits Notch signaling, and it's the function of RBM10 along with probably some other proteins that maybe have the same effect to induce exon 9 skipping. So when you have more RBM10, you make more of the exon 9 skipped version, which leads to more inhibition of the Notch pathway. So now we know everything there is to know about how alternative splicing can regulate genes that are involved in cancer, right? Yeah. So it turns out that there's a gene called SF3B1 that when we look at RNA processing related genes that are mutated in cancer, SF3B1 is by far the most frequently mutated gene. Mutations in SF3B1 don't change the alternative splicing of cassette exons or alternatively included exons. Mutations, when people look at tumors that have mutations in SF3B1, they find that instead of having cassette exons regulated, those genes leave an extra piece of intron in the messenger RNAs. They utilize an alternative 3' splice site. But this only seems to happen in messenger RNAs where that particular intron has a fairly poor, fairly difficult sequence for the spliceosome to recognize. What happens is that when SF3B1 is mutated, its interactions with other components of the spliceosome are disrupted. And what that means is that it can no longer displace a protein called SF1 from the branch point where the lariat is supposed to attach to. And therefore, a protein called P414 along with SF3B1 goes looking at regions upstream of that region to see if it can find something that kind of sort of looks like a splice site. And if it does, it includes a chunk of the intron into the finished messenger RNA by splicing to an upstream cryptic splice site. It turns out that many of the genes that are misspliced when SF3B1 is mutated, because they have these sort of not very good splice acceptor sites are genes that are involved in regulating cell division and apoptosis. Although at this point, there's no poster children that I can tell you about where we can say, oh, here's exactly why this particular missplicing event leads to abnormal cell growth. It turns out that NUM is definitely not the only protein involved in regulating cell division and apoptosis that is in turn regulated by alternative splicing. So BCL2, FAS, Harvey-RAS, cyclin D1, caspase-2, all of these are pro-apoptotic or pro-cell division genes that are known to be regulated by alternative splicing and to produce different forms that either are more capable of promoting cell division or more capable of encouraging apoptosis. So we've talked about how there are different mechanisms through which alternative splicing can be regulated, leading to the alternative splicing of cassette exons or alternative exons, and that sometimes that alternative splicing simply means that the splicing machinery chooses different places to start splicing an intron and utilizes different 5' splice sites or 3' splice sites. And we've also talked about how many of those genes that are regulated are genes that are involved in cell cycle regulation. The pretty color diagram at the right shows additional genes that are known to play important roles in regulating cell proliferation and apoptosis. And the reason I'm showing you this is that there are examples of all of these types of splice regulation that lead to the production of different versions of proteins, or in some cases probably no protein at all through nonsense-mediated decay by alternative splicing. So FGFR2 is regulated by using alternative exons. BRCA1 involves the utilization of cassette exons. And there are also examples where, for example, DMTF1 utilizes alternative 3' splice sites, including different chunks of this intron, where KLS6 utilizes different 5' splice sites and includes more or less of this exon. So there are many, many different examples, and in the case of many of these proteins shown in this diagram, we know that the different versions of the proteins that result from this alternative splicing have different effects on cell proliferation and apoptosis. Finally, to put the cherry on top, many of us are interested in understanding exactly what RBM5, RBM6, and RBM10 are doing and how that relates to tumor progression in cancer. When we look at alternative splicing that's regulated by all three of these different proteins, we see examples of not just cassette exon regulation, not just alternative exon regulation, but also regulation of the utilization of 5' and 3' splice sites. And while we've been talking about machinery that regulates cell proliferation and apoptosis, there are other mechanisms that are important for tumor progression, for oncogenesis to occur. There are other pathways that are regulated or disrupted in tumor formation. And it turns out that alternative splicing plays an important role in regulating genes in a number of these other pathways as well. So hopefully after watching this video, you have the idea that regulation of cell division and apoptosis through splicing is complicated. Some things to keep in mind. This video up till now has suggested an air of confidence and knowledge about this process. That's probably kind of a lie, because We probably don't know most of the genes that are regulated as part of the process of abnormal cell division in cancer, as part of the process of interfering with the process of programmed cell death, that are regulated by splicing. There are probably a lot of genes that we simply don't know they exist, and we don't understand their role in cancer. Of the genes where we know how splicing is regulated, if that change in splicing basically causes the protein to go away because it messes up the messenger RNA so that it's degraded through nonsense-mediated decay, we have a pretty good idea how those things work. In the cases where, as we saw, like for NAM, where the exon 3 or exon 9 are skipped, in the case of NUM, because we studied the heck out of it, we have a really good idea of what those changes in the structure of the protein do. And even there's not complete agreement among scientists about exactly how those things work. In the case of just about every other example of the proteins I've shown you, and the ones that we looked at yesterday where we could see many, many different alternative splice forms, we really don't know what happens when you remove those bits of protein and why that affects or how it affects the function of the protein. And finally, in cases where we do see changes in the splicing of a protein that serves as an oncogene or a tumor suppressor, I would say in every case, we don't really understand all of the proteins that are regulating the splicing of that particular gene and how that process is happening. So there's a lot of unknowns, a lot of things we still need to know.